So I thank the organizers for this kind invitation to that gorgeous meeting here in Antalya. And just to show you where I work, Central Institute may not so well known. It's in Mannheim. We are affiliated with the University of Heidelberg. And actually, this institute is in Mannheim is laying actually right in the middle of the Turkish community, most of the people, of Turkish people living around here. And so we also seeing a lot of patients from that area. It serves the entire Mannheim area of thought. So that's our, this complex here is the Central Institute, and we have the fortunate situation that we can couple clinical and preclinical research very tightly. So this is where I live, this work half-time. The Department of Psychopharmacology, and this is the other clinic, the addiction clinic, up here in this corner down here. So then I have to obviously make my disclosure. I don't have anything to disclose, and for those who have to go for competing interests here, that's the short version of my talk. I will going to talk about therapeutic development for alcoholism targets within three major systems, which either mediating reward, stress, or executive control. And I will try to put a translational perspective on here, first on the pharmacogenetic effects of opiate-dopamine interactions. Then I will move on to the stress system, how we can try to target that. And that there is evidence for hyperactivity of stress systems in alcohol abstinence. And I will then make remarks to altered glutamatergic transmission in corticoaccumbal circuitry as key mechanisms for relapse and addiction. And if you think that these targets are highly overlapping with other areas of psychiatry, like depression or ADHD, you are completely right, because they do. And so what's the problem here? Coming from Germany, I have to show you some pictures from the Munich Oktoberfest, and these two chaps actually represent very nicely what we love about alcohol, either the sedating or the activating effects of the drug. Then what happens is, of course, we tend to drink too much of it. But that's not only a human problem. So that's actually, alcohol is a commodity. We really drink too much. It's mostly in the developed country, but the developing countries are catching up. This is the world map of alcohol consumption worldwide, and you see it's a big problem in Europe, an even bigger one in Russia, but also in the United States and Australia. Then I don't know about Turkey. It looks very bleak here, but I think it's also a regional problem, because actually it turned out alcoholism very often goes with affluence. As more richer the people get, as more they tend to drink. So, as I said, it's a commodity. And why do we like it so much? It's, you can see that already in the animals. And actually, you know alcohol is a natural resource, typically only up to 1% of alcohol content available. And here we found in a study some years ago a little ecosystem where you have exceptional high concentrations of alcohol, up to 4%. That's average beer probably. And there's this little mammal here, which actually is more closely to us than to the rats. It's a tree shrew, and which his pen-tailed tree shrew, he pollinates these palms. And during this time when these palms are flourishing, that animal lives exclusively on this nectar, on this 4% alcohol, which it means it drinks more than a bottle of vodka per day. So here also some data what we did with these little animals. You can basically actually demonstrate that they indeed go for the nectar with the alcohol. You can give them some nectar without alcohol, they won't like it, and that's field psychopharmacology. That was not done in the lab. And if you then compare here, you can also measure not blood alcohol levels, but some biomarker for alcohol, which you, is this thing working? oops. So it's basically the gray shaded area is what alcoholics typically take, and the big bar is what the tree shrew is taking during these several weeks or up to a month of when these fruits are ripe. So he drinks much more. However, he doesn't show any signs of decreased fitness. So there's no intoxication, there's no getting catched by predators or something like that. So somehow this animal deals with it. And unfortunately, we can't. We did obviously two forward development happened. We learned to make much higher alcohol contents, and it became much more available, which then at the end tends to alter behaviors. And here you see what starts to become the problem. We set aside our so alcohol does something specific here, which other natural rewards don't do, even in moderate consumption. And I show you here this piece of data from Patricia Yanak's lab from the Gallo Institute in San Francisco. And what they did, basically they learned rats to press a lever for alcohol. And you can do that, you know that, the Skinner boxes, and they do that happily. And then, of course, you can introduce a devaluation of the alcohol by just saturating the animal before. So you let it drink freely without pressing a lever, and then you put it in the operant box. And what you see here, what will going to happen in this graph where it says devaluated, of course, the animals don't work so much for the alcohol anymore. In the first week, in the second week, but after 4 weeks and after 8 weeks, that difference is not visible anymore. So the animal doesn't care if it has been drinking before, and these are moderate amount of alcohol. They never intoxicate, so they never reach a legal limit or something like that. They don't show signs of intoxication. Yet, they still work for the alcohol, take it even if they had taken it just before in their home cage. And here on the bottom, you see some neurocircuitry which could mediate that. The bottom line is that the dorsal striatum may mediate this process. If you lesion the dorsal striatum or inactivate it, you don't see this process of habituation and automated behaviors. This is with alcohol doses very low, as I said, after a pretty short period of time of chronic consumption. In humans, this leads to very different problems. David Nutt made an overview about the effects of what drugs are doing to you or to others around you in the UK. This is kind of 2010 data. And obviously the normal subjects are showing up. They all do some harm to us and to others. But the real outlier here, which is doing most harm to us or to the individual and to the others, is alcohol. So they basically classified as alcohol as the most dangerous drug for our societies. And when you see this, around that time there was a British lady that was before Amy Winehouse in the press, which made it within the year of 24 to die from alcohol, not from intoxication, just from liver failure. And the British scientists actually think that within a relative short period of time, alcohol will make a major impact, even more than now, and will become number one of mortality, so surpassing cardiac disorders. So what do we have? So what is addiction? You all know that. I wrote a little different version of the definition. What we have in addiction is a very strong attentional bias. We have a chronic negative effect when access to the drug is prevented, and that can be voluntarily by abstinence or just when you can't get it. Then we have a loss of control over the drug, and then there are these kind of phenomenon of unresistible urges called craving. So how can we treat this? So this would basically preventing this is the main clinical challenge, not treating your withdrawal syndromes. And there's this famous review nowadays, and I still like it, from George Cooper and Nora Volkoff, delineating 3 major systems which may mediate these craving or these predicting or precipitating relapse. And obviously it's the mesolimbic reward system, but it's also stress systems and its executive function and inhibitory control. What do these systems actually do? The mesolimbic system obviously type of gives a go signal. Then the stress initially is kind of a warning signal not to take too much of it, but it will run astray and finally be a major precipitator of relapse. And then, of course, the cognitive functions, let's stop and think about it a little. That's what we see is also heavily impaired, and that's probably at the core of the problem. So what do we have to target these things? Obviously, the reward system is all about the dopamine-opiate interactions, and what the drugs are there, these are the opiate antagonists, and I will come to it in a minute, why the opiate antagonist. I won't go and show any meta-analysis, but here we have meta-analysis from 10,000 and more patients for naltrexone treated, and we can calculate numbers of patients treated for one successful outcome, the numbers needed to treat. It's around 6 to 7. So there is no big efficacy with these drugs. There's no magic pill or magic bullet available here. So, and people have started thinking about this heterogeneity. So what's the underlying problem here? And maybe some of it may in fact be due to genetics. So just to recap for those who are not in the field, what have the opiates to do with the dopamine? They provide an inhibitory drive on the interneurons in the VTA, and if that alcohol releases these endorphins, and by that leads to a disinhibition of the dopaminergic system, giving us more dopamine in the target regions. Then a while ago, some studies in monkeys showed that there's a big difference between monkeys, how they take alcohol when you treat them with an opiate antagonist, and that's basically by gene. There's a certain variant in the mu-opiate receptors, which here on the left side of the graph you see these two groups of monkeys according to their genotypes, the ones taking more alcohol but responding very strongly to naltrexone, while the others taking less alcohol but don't respond to the naltrexone at all. That was then followed up by our group at the NIAAA, led by Markus Heilig, with a PET study where we showed that if you give alcohol, you can displace the radioactive tracer raclopride, the D2 antagonist, and by that displacement you can basically have a surrogate of dopamine release. So if the bars are going down, dopamine is released. And as you can see here again, that's by genotype. So you either release a lot of dopamine or little. And that's a healthy subject. That has indeed a clinical impact. When you then stratify, for example, a large American treatment trial, the combined study, for the effects of this genotype, you actually see that the opiate antagonist only has an effect in one of these genotypes, while the others are not responding. So we followed up on that by generating a humanized mouse who exactly carries this genotype, this human single variant, and you see here in a microdialysis study, which is basically Pangang to the healthy subject dopamine antagonist displacement study, that by genotype you release more dopamine. And when we then treat these mice with either of the available opiate antagonist, we see that the efficacy of the drug is much bigger in the variant allele. So here's one reason for the heterogeneity of the treatment outcome, maybe by genetic effects. However, there are other things which are not so really clear about it. So we put some rats in an fMRI scanner and did the resting state fMRI to assess functional connectivity. Functional connectivity means how region in the brain talk to each other based on their spontaneous fluctuations. And you typically see these kind of correlation matrices here on the left, which don't say you so much. And then you get some activation maps in certain areas. But you can do a much better job by applying graph analysis to that to try to figure out what actually every of these node or region in the brain is participating in the communication. And you see, you can identify certain features here. Some are more in the center of the communication, some are more in the periphery. I won't go into the details here. So what we did then, we let rats drink for a while and assess their connectivity according to this graph analysis, and you can see strong changes in different regions, in different measures of how they communicate to others, what's their role in the communication network. And the important message here first is there's no ventral striatum, no nucleus accumbens in that, where actually we all thought would be the action of naltrexone in particular to look for. More importantly, when we let these rats have be 1 week abstinent, there's no really changes in these already altered network properties. But if we give them a naltrexone, we basically see a little bit pushing these network properties towards back to normal. So everything, not everything, a lot what is blue in the alcohol condition got now red in the naltrexone condition. So naltrexone seems to do some good indeed, but if it is really via the opiate-dopamine interaction or via a complete other mechanism on cognitive processes or cortical structures, we don't know. So there's a lot to be learned still about naltrexone. Okay, then let me talk a little bit about the stress systems, which gave us a lot of grief. They actually lead to hyperreactivity of addicted persons to stress, are involved in withdrawal and anxiety. There are a lot of treatment trials which have been done. Odanzatron, probably the most successful, is an 5-HT3 antagonist, but the others were actually more rather a problem. Let me go into this a little bit from the data from the rats. So we have a possibility to make rats truly alcohol dependent, and I won't go into the detail for that here, but the outcome of that, there's a long-lasting change in behavior, even in the absence of the alcohol. And so we can do that in this cartoon. So we treat these animals with this alcohol for several weeks. They go through multiple withdrawals and they feel more and more worse. And if you then do a CVR score, which is an assessment for patients for the withdrawal scores when alcohol is discontinued, you got a true difference here. So these guys are dependent. The important thing here is what's the long-term outcome of these. So we call them post-dependent because now they don't get any alcohol anymore and are 3 weeks in their home cages. And if they get a little bit stressed, they react very different than normal rats. They hyperreact. And you can see that here in this little graph, which would be how much alcohol they drink when they get a stressor, like a forced swim stress. And they, of course, do that take much more alcohol than the controls. But what they also show is an upregulation of extra hypothalamic CRH systems, here displayed by the CRHR1 receptor in the amygdala. Based on these data, the NIAAA lab had then done a treatment trial, an experimental medicine trial, for the neurokinin R1 receptor, and the neurokinin R1 system is probably the CRH light system.
Mr. Sommer, last 1 minute.
Yeah? Okay, it's the last slide. I can go finish with that. And so they see a lot of they see beneficial effects by here depicted on the on that imaging study that alcoholics have without treatment and hyperreactivity to IAPS, also emotional stimulating images, negative stimulating images, while with the substance they indeed improve here and even get more responsive to positive signals. However, The recent paper from this group was very sobering here. The trial failed after many years, and what they showed is they indeed got hand of a CRHR1 antagonist, and the effect were negative on the outcome, and their conclusion here was our results may indicate that antagonists with slow offsets are required for therapeutic efficacy. Alternatively, the extensive preclinical data on CRHR1 antagonism as a mechanism to suppress alcohol seeking may not translate to humans. And that actually illustrates very nicely the problems with translating data from animals to humans. And with that, actually, I thank you for your attention. I can run forward to my thank you slide.